Hi everyone, I am Elisa Corti and I am the early stage researcher number 7 of the European founded consortium SimpleSci. I am Italian but I am based in Coimbra, a city full of color and music in the middle of Portugal, that hosts one of the most ancient universities in the world.
Here in Coimbra, I work at the Center for Neuroscience and Cell Biology under the supervision of Professors Carlos Duarte, Paulo Pinheiro and Ramiro Almeida. All of them study the interaction between neurons from different points of view. Indeed, the goal of my project is to define the role of a protein called FMRP in the process of communication between neurons, and how alterations in this protein are linked to disease. But let's start from the beginning. These are two neurons, the cells that are found in the brain. If you look, for example, at the red one, you can easily see a long thin branch called axon, other shorter branches called dendrites, and the main body of the neuron called soma. Neurons can talk to each other, and thanks to this, the brain can work properly and control many aspects of our life, from cognition and memory to sensations and body movement. This happens in a specialized structure called synapse, in which the axon of a neuron is close to the dendrite of another neuron. But how does this communication work? When a neuron wants to say something to his neighbor, it sends a signal along the axon. This leads to the fusion of the so-called synaptic vesicles with the cell membrane, and to the release of molecules that act as a messenger, the so-called neurotransmitters. These molecules then bind to specific proteins on the listening neurons, called receptors, with the subsequent activation of this cell. Synapses are composed by a huge number of proteins that cooperate to make everything work smoothly. If any of these do not work correctly, neurons are no longer able to communicate in a precise way. And these may cause different neurological conditions, generally named synaptopathies. The main aim of the consortium, which I am part of, is to study the role of synaptic dysfunctions in neuropsychiatric disorders and to find the connection between such dysfunctions and the symptoms observed in patients. Nowadays, there is no cure for these conditions, so it's important to understand what is really happening at the synaptic level, to find the new therapeutic targets and to try to ameliorate patients' life. In my project, I'm focused on fragile X-mental retardation protein, that I will call FMRP, and which is involved in the regulation of many synaptic proteins. The absence of FMRP leads to Fragile X syndrome, that is the most common inherited form of intellectual disability, and it affects 1 out of 4,000 males and 1 out of 8,000 females. This is a broad disorder characterized by many symptoms such as intellectual disability, stereotyped behaviors, anxiety seizures, and sleep disorders. Synaptic dysfunctions have been previously observed in mice models of this disease, but the relation between the absence of FMRP, synaptic dysfunctions, and symptoms is still poorly understood. To study this relation, I will look at neurons that have low amounts of FMRP, and I will compare their synapses with the ones from neurons with normal amounts of FMRP. I will focus on 3 different aspects. First of all, I will use microfluidic chambers to separate the axons from the dendrites and some of neurons, and I will study if the absence of FMRP influences the axon development. Then, I will study the process of vesicle exocytosis and I will use autoptic cultures. Here neurons can talk only with themselves, so I will be able to listen to what a single neuron says without listening to any other cell. Finally, I will study how the absence of FMRP affects the presence and function of neurotransmitter receptors thanks to quantum dots, a technique that allows me to track the movement of the receptors on the neuron surface. Presently, there is no cure for Fragile X syndrome, but hopefully understanding what is not working in the communication between neurons will help discover new therapeutic targets, or at least improve the course of the disease.
I hope you guys enjoyed this video, and if you're interested in the topics of our research, check the SimpleSci website. Also, don't forget to follow us on the social media, so Facebook, Instagram and Twitter. Bye!